So this topic was already introduced today to a degree by Dr. Zhang from Tiantan, and so I think it's agreed that we don't have optimal treatment for this disease. It's probably the worst cancer in children. That's diffuse intrinsic pontine glioma, or DIPG. I'm going to tell you the results, early results from a phase I study that we launched. It's a first in the world study, has never been done before. And it's the first time I'm presenting these data outside of North America, and the first time, of course, here in China. There's the title of my talk, and I just want, at the very beginning, I want to acknowledge the research team that has helped me with this particular study. And these are the researchers from graduate school, from other departments nearby across the city that were very involved in this particular study, because it involved a huge group to orchestrate and to organize this particular study. So there's the title once again, you can see. And what is DIPG? So DIPG is a tumor confined to the pons. You can see the signs and symptoms here. Age is young, usually age 5 to 7. Hydrocephalus late in the course of disease. There's a role for biopsy I'll talk about. There's no role for complete surgical resection here. And radiation fails, chemotherapy fails. Nothing seems to work in this particular tumor. In the last 5 years or so, we've understood the molecular genetics of these tumors. And you're seeing, these are all studies that were published the same year in Nature Genetics, the same issue in 2014, illustrating the role of the ACVR1 mutation in this disease. And so this becomes a target then for DIPG. I mentioned biopsy at Sick Kids Hospital in Toronto. What we're doing now is stereotactic robotic biopsy. I usually choose an approach from the posterior fossa, so coming down the middle cerebellar peduncle. I usually stay lateral. There was a question earlier about where do you biopsy. Usually stay fairly lateral, because biopsying central, there's so many crossing fibers, there's so much risk of injuring important pathways. So I stay lateral. Usually these tumors don't enhance, but if there's an area, a small area of enhancement, I would target the enhancing area, but usually they do not enhance. So you can see you can get biopsies from these, and they can be very useful for determining what the molecular genetics are for these particular tumors. And almost all of them have mutations in the H3K27 histone modification gene for these midline gliomas. And they can occur in the brainstem, in the thalamus, and sometimes elsewhere. But largely they're midline tumors, and by and large they're all malignant tumors. So I was very proud that I was able to publish this work, this review, in the Chinese Journal of Neurosurgery. So thanks to the publishers and the editorial board for allowing us to publish this in the Chinese Journal of Neurosurgery. But everything you need to know about DIPG is on this slide, including the genetics, the H3K27 mutation, the MYC proto-oncogene, and some silent pathways. On the bottom you can see. And then all the clinical pathological features and current treatments and mutations are all listed on this slide. So you can refer to this article in the Chinese Journal of Neurosurgery. Radiation has failed. I think I'll just leave it like that. We tried all different types of radiation therapy, from hyperfractionated to radiation sensitive. Nothing works. Proton therapy fails, photon therapy fails, gamma knife fails. Nothing works. And it's not for lack of trying with chemotherapy. So you can see here from this website, DIPG.org, now almost 100 clinical trials are ongoing for this tumor, and we hope something is going to work, but previously so much has failed for this disease. To think about convection-enhanced delivery, and Mark Swadan's a neurosurgeon in New York City, and he's tried this technique for DIPG with some success in some limited cases. But we chose to use MR-guided focused ultrasound because these are non-enhancing tumors. The blood-brain barrier is intact, so we asked the question, can we overcome the blood-brain barrier by using focused ultrasound in this disease? So we did some basic preliminary lab work. The picture is of Saira Ali. She's a PhD student working in my laboratory, and she showed that using focused ultrasound does enhance the delivery of a chemotherapeutic, that's doxorubicin. I just want to spend a minute on this schematic in the bottom left-hand corner because it shows our mouse model, and what's important to know that this is low-frequency ultrasound, not high-frequency. High-frequency ultrasound is used for lesioning like you would use for essential tremor, for Parkinson's disease, and other types of lesions in the brain. Low frequency does not create a lesion, but what it does do is allow you to use microbubbles, and you can see in the bottom schematic, the microbubbles in the presence of focused ultrasound will expand. They'll spread the endothelial cells slightly apart to break down the blood-brain barrier, allowing chemotherapy and drugs to pass more readily than can happen without opening of the blood-brain barrier. It's transient opening. It stays open for about 4 hours and then it closes. We did a rapid high-throughput drug screening, and we looked at many different drugs, and the drug that was most effective against DIPG was doxorubicin, and that bar graph is showing you the concentration in the brainstem compared to elsewhere in the brain. So we were able to concentrate it in that area of the brain. And this is the workflow in an animal model showing how we use MR-guided focused ultrasound with reproducible targeting and delivery of doxorubicin right on target, right into the brainstem in a very small animal model. We were able to show that we had tumors that we were able to create that were very similar to DIPG in humans and able to show contrast enhancement, and they were highly infiltrative and able to target those with MR-guided focused ultrasound. And then showing that doxorubicin, which is a drug that's almost never used for brain tumors because it does not cross the blood-brain barrier. It's used for sarcoma, it's used for other hematologenous type malignancies, but almost never for brain tumors. We were able to show that we could decrease the proliferation in the Ki-67 index using doxorubicin following focused ultrasound and microbubble treatment. This is work that was done by a Japanese postdoc in my laboratory, Dr. Joji Ishida, who published this work recently. So this led to the description and also the writing of a clinical trial for us in this protocol. And we have a relationship with industry, with Insight Tech. They're a company from Israel with whom we collaborate, and they have provided the MR-guided focused ultrasound unit for us to perform this study. On the right-hand side is the cycles of chemotherapy that are used and how this clinical trial is embarked upon. It's a very labor-intense clinical trial, as I'll show you. So we treated our first patient about a year ago, and it required a huge team of professionals. So we had anesthesia, neurosurgery, we had the oncologists, the chemotherapy nurses, we had medical biophysicists, we had MR technicians, everybody, about 18 individuals helping with the treatment of this first patient in the world. There you can see the application of the head frame. So this is done under general anesthesia. In the middle figure, you can see the diaphragm goes into the water bath. We need to cool the scalp with the water bath so that the focused ultrasound doesn't cause any heating. We do not need to shave the hair. But look on the right-hand side, all the people that are involved in this particular study treating this first patient. So general anesthetic, head frame, water bath diaphragm, transport to the MRI scan, microbubbles, and following the microbubble administration and focused ultrasound, the drug is given because the blood-brain barrier is now open, and we can see the results. So what did we see? This is the first patient, the first treatment, 50% of the tumor we targeted with the first treatment. We then moved to 75% of targeted treatment a month later, and then finally we treated the whole tumor volume 1 month after that. So you can see these are non-enhancing tumors. This is an enhanced scan on the far right, and the whole brainstem is essentially enhancing, but it's enhancing in a capillary-dependent manner. So it's more physiological to get this type of enhancement in this type of distribution than I think it is with convection-enhanced delivery. Here's an example of the second patient treated. 75% of the tumor treated. You can see on the left-hand side the coronal image in the middle, and then on the right-hand side 24 hours later, we do an MRI scan to make sure that the blood-brain barrier is closed, and you can see that there's no enhancing brainstem or pons on the right-hand side. So we've now had an opportunity to treat over 5 patients on this phase 1, so it's a feasibility safety clinical trial for DIPG. And we are collaborating with another center in Washington, D.C. in the United States, and they've also entered patients, so we're accumulating a lot of experience now. I can tell you so far, it's very preliminary, that patients have all tolerated the procedure. There's been no hemorrhage in the brainstem, there's been no bleeding consequences, no edema, no swelling. These patients wake up very rapidly after their anesthetic and go home the next day after their MRI scan. None of the children have shown any side effects. So just in summary, this is a universally fatal tumor by and large. There's no rule for aggressive resection. We can talk about the need for biopsy. I use a frame and robotic-based methodology for that. Conventional radiation therapy, I should mention that these patients are treated MR-guided focused ultrasound after radiation therapy has been given. Targeted chemotherapy may be coming down the road subsequently, and then we'll need additional trials, as this is the first platform that we're using for MR-guided focused ultrasound. Future directions, here you can see a biopsy on a patient with DIPG, and this is almost real time. So within 2 weeks of diagnosis, we can get this type of readout with various pathways that are involved that could be treated by active drug therapy. And I'm listing here all the different drug pathways that could be targeted for with MR-guided focused ultrasound to increase delivery to the brainstem. The goals of future therapy for DIPG, in my opinion, will be getting early diagnosis with genetic characterization, possibilities of doing CSF and blood typing and circulating tumor DNA analysis as opposed to doing open biopsy or stereotactic biopsy, and then treating up front before radiation therapy. Imagine if we could do that, and then reserving focal radiation therapy later if we needed to. We're still many years away from that, but I think that's where the future is going to be. And we're already talking about using MR-guided focused ultrasound with immunotherapy approaches, CAR T-cell therapy for DIPG in the near future.